Welcome back, guys. To pick up where we left off, remember that yesterday we discussed this word called homeostasis, which meant relatively stable internal conditions. That's basically the goal of all cells and organisms, is they want to keep their conditions similar. We talked about how cell membranes were part of that, part of how a cell could keep its conditions stable. So today we're going to talk about how water and substances can move across a structure like a cell membrane, and some different ways that cells are able to get materials in and out of that cell. Okay, so first we're going to start with something called passive transport. So if you're a passive person, you don't actively do things. So passive transport doesn't require energy. The cell doesn't really have to do anything special to make this happen. Okay, so molecules move across the cell membrane. If you're taking notes, make sure you put down that it happens from high concentration to low concentration. Okay, it's probably going to be on here later, but that's how passive transport works. So there's two main types of passive transport in a cell. We have diffusion and we have osmosis. So what's going on right here, this is definitely diffusion. There's not nearly as many of whatever those little circles represent out inside the cell. So they can move from outside to inside because it wants to achieve something called equilibrium. Okay, so diffusion, like we just said, molecules move from high concentration to low concentration. That's what it is. And osmosis is the diffusion of water and only water. Osmosis is the diffusion of only water. So if you're taking notes, write that down. I can promise you're going to have to know the difference between those two on your unit test. Okay, so there's three types of solutions, and all of this is relative, which means that we have to realize what way we're comparing things to. So we're usually comparing the inside of the cell to the outside of the cell. So these are the types of solutions I can put this cell in, okay? So isotonic, the concentration is the same on both sides. I don't know if you remember this from like middle school geometry, but there's something called an isosceles triangle where both sides are equal, like the bottom side's smaller, but these two sides are equal. So that's isosceles. So iso is like equal. So isotonic, the concentration is the same on both sides. So equal amounts of water enter and exit the cell, so its size stays constant. That's that equilibrium word that's going to come up. So then I can put a cell in a hypertonic solution where the concentration is greater. So if I put a cell in a hypertonic solution, outside's hypertonic, inside's hypotonic. So it's going to shrink. And what you see in picture number 2 is actually what happens to a blood cell in a hypertonic solution. This would be like if you put it in a really concentrated salt solution, or if you put it in a really concentrated sugar solution. It's trying to get the same concentration, like the same amount of sugar per milliliter of water. It's trying to get that same balance. So it'll lose pure water out of it just so that it can be more concentrated. And then hypotonic, it takes water in, so it puffs up. So hypotonic, the concentration is less than the other side. So like if something's hypoallergenic, then it has only a little bit of stuff that causes allergies. So it's lower. Hypertonic, like hyperactive, you're too active, right? So isotonic, hypertonic, hypotonic. You need to know the difference between these three. And you also need to know what happens to a cell when it's in each of these solutions. So you might want to pause this video and draw a little diagram, make some notes to yourself. Like I said, you're gonna need to know how to do this. Okay, here I wrote it down. So isotonic, cell stays the same. Hypertonic, it shrinks and water leaves. And hypotonic, cell swells and water enters. Actually, in number 3, a cell can pop. So a fun fact, especially if you are taking medical terminology or any of those like nursing classes, there's a reason why when you go to the hospital, they don't just put pure water in your IV. It's always saline. And that actually happened to me once. Like, I got super dehydrated because of an illness I had, and I had to go to the doctor, and they had to put fluid in my arm. It wasn't pure water, it was saline. And not only is it salt water, but it's salty to the exact point that blood is supposed to be salty. Because we want our cells to look like number 1. If you do pure water, your cells look like number 3, and they can actually pop. And you die this way. There's a case study where this happened to a woman. She needed an infusion of fluids, and they went to the pharmacy in the hospital, and they got pure water and put that in her arm, and she died before they could save her. So that is a problem. Okay, so there's also something called facilitated diffusion. So a facilitator is someone that helps something happen. So this means that diffusion is happening without energy. It's just I opened a door in the cell membrane so that other things can get through. This is where those protein channels from yesterday come into play. We said yesterday that we want to get things in. We need to get sugar in or bigger molecules. We have to. The cell needs them to survive. We just don't want them to come through the membrane all the time. So we can open up a channel or a transport protein that will let those things through. So for instance, I can't just Kool-Aid man my way through the wall. Okay, I'm not that good. But if there's a door open, I can walk through it without a whole lot of effort. Like, the wall doesn't have to do anything special to get me through. The door's just open. Okay, so let's talk about what happens when we need to use energy. So we talked about passive transport just now, like diffusion and osmosis and all that. It doesn't require energy. Active transport does. So active transport is powered by chemical energy, or ATP, and occurs through transport protein pumps. So even if I dig a hole in the ground, I'm not going to get oil and natural gas out usually. I usually have to pump it out. So that's what we're seeing with active transport. So cells use these to maintain homeostasis, especially if I need bigger molecules, say sugar. Sugar is actually pretty big. I need sometimes to actively get it. Sometimes it's even bigger molecules than that, like hormones or things. I have to pump those inside. Or maybe it's a type of particle that is charged and can't get through the cell membrane. All right. So let's talk about this diagram for a second. So what can happen is, where you see number 4, a molecule of ATP can attach to that channel, and it can pump things out of the cell. I use this to go against the concentration gradient. So if I want to build up more stuff in the cell than is outside the cell, which your muscles do all the time, they can pump sodium or potassium in by attaching energy and like using energy to pump it in against the concentration gradient. So you see it up top, that active transport in bold, write that down. Active transport requires energy input and enables the cell to move a substance against its concentration gradient. So I can go from low concentration to high concentration, which is the opposite. Okay, so sometimes if I'm a cell, I can't just wait for diffusion to do things on its own sweet time. Sometimes I gotta be proactive about this. So let's say I'm a cell and there's a whole big cluster of, say, sugar or dissolved proteins or something that I know I need. So there's something called endocytosis, because endo means inside, where the cell can just engulf or take in a whole bunch of stuff at the same time, or something big. Your blood cells do this all the time. So there's something called lymphocytes, or what's the word? The white blood cells, okay? Macrophages, that's the word I'm looking for. So there is a big type of white blood cell that it bumps into a cell that doesn't have those right protein markers on it, it will eat it. it'll just slowly fold its cell membrane around it and eat it. And remember, bacteria are super small compared to cells, which you guys saw in the picture for this chapter, you know, that big booger looking thing with the little red things on it. That's a big cell that could eat those bacteria. Not all of them do, but some of them do. So phagocytosis is a type of endocytosis. So that's what's going on here. It found a bacteria or a whole bunch of something at once. It can fold the cell membrane around it and create a vesicle. Or remember, vesicles are just like storage pouches in the cell. Okay, so then the vesicle fuses with a lysosome and the enzymes break it down. So that's how I'm digesting it. We said lysosomes were like the cleanup crew in a cell. So in this instance, it ate a bacteria or it ate like a big chunk of something that it wants to digest. Those lysosomes can digest it for it. Okay, so exocytosis is the process of expelling material from the cell. So if I am a cell that produces, say, adrenaline and something scary happens, like the fire alarm goes off and it hurts and I'm panicking, right? So exocytosis happens when those cells are making a whole bunch of adrenaline to try to get me that get up and go. They can't wait for diffusion to just lazily diffuse those out. It needs to pump out that adrenaline. I gotta go, okay? So instead of just doing one molecule at a time, it'll send out like a whole cluster of those and push them out of the cell. So exo means outer, and we're expelling things out of the cell. So it's just the opposite of what happened with endocytosis. The vesicle is made out of the same stuff as the cell membrane, so it can fuse with that outer membrane. So we see it's transporting to the outer membrane, it's attaching to it, and then the membrane fuses with the cell membrane and releases its contents. It just dumps it outside the cell. There was one thing these slides didn't mention. There's something called, we talked about phagocytosis up here, so I'm going to go back. So we talked about phagocytosis. There's also something called pinocytosis, P-I-N-O, cytosis. And that's where I'm absorbing like something that's dissolved in liquid, phagocytosis. I think like fat kid cytosis, that's cell eating. Anything with that P-H-A-G means eating. So that's what's going on there. Yeah, it was a little scattered, but I hope you guys were able to get a hold of this. You are going to need to know how to tell what direction water's going to move. There's going to be some of that as you finish your web quest. And this weekend, you'll also have a practice assignment on this because I think that's one of the hardest things for students to grasp when it comes time for this unit test. That's one of the most commonly missed things. So make sure you are focusing your efforts on learning how things will move. Like water moves from where there's a lot of water to where there's a little water. So just make some notes to yourself, sketch it out. Let me know if you have any questions, and I'll see you guys in the next video. Take care.